All right, so continuing on with chapter 40 or chapter 50, we're going to start talking about taste and smell. In terrestrial animals, both of those are going to rely on sensory receptors. Gustation taste is going to detect chemicals called tastants, and olfaction smell is going to depend on detecting odorant molecules. These both use chemoreceptors. Aquatic animals are not able to distinguish between taste and smell. Insect taste receptors are found in sensory hairs called sensilla, which are on both their feet and in their mouth. In mammals, taste receptor cells are in modified epithelial cells, aka taste buds, that are located in several places. Sorry about that, the grammar, both in your tongue and your mouth. Five taste perceptions: sweet, sour, salty, bitter, and umami, which is elicited by glutamate. Receptors for each of these, except salty, have been identified. Um, Researchers believe that individual taste cells are able to express one receptor type, but they can detect, and detects one of your five tastes. Regions with taste buds, any region with them, will be able to detect all of the five types of taste. So, taking a look at your tongue, each of those individual little dots that are on your tongue are papilla. Those are going to lead to your taste buds. And then within your taste buds, again, you're going to have your sensory neurons that are going to recognize each of those different taste and then be able to send that signal forward. Olfactory receptors are neurons that are going to line your nasal cavity, the upper portion there. These molecules are able to bind to, when they bind to receptors, will trigger a signal transduction pathway, which will send action potentials to your brain. Humans are able to distinguish between thousands of different odors. These both have different pathways, but the senses do interact with one another. So there's your odorants moving in to the nasal cavity. As the odorants move in, you can see that there are different receptors depending on the odorants that are present. Once they have bound to the receptors, again, they're going to send off that signal transduction, which is going to generate the action potential, which will then move into the central nervous system. Protein filaments are required for muscles to be able to move. Muscles move as a result of an input into the nervous system through one of your neurons. Action of a muscle is going to be to contract, while extension will be passive. Skeletal muscle, also known as striated muscles, what moves your bones and your body. Lots of smaller units work together in concert, form a hierarchy. We have skeletal muscles that are made of long fibers that run parallel to your muscle's length. Each of those individual fibers is made of several smaller myofibrils. Those are arranged longitudinally, and they consist of two kinds of myofilaments, both thin and thick. Your thin filaments consist of two actin strands and two strands containing regulatory proteins. Thick filaments are staggered arrays of myosin molecules. Again, that's what's going to give you that striated pattern. It's got a regular arrangement of these, light and dark bands. The functional unit of skeletal muscle is a sarcomere. It's bordered by what we call Z lines, which are where the thin filaments are attached, and they project towards the center of your sarcomere. The thick filaments are attached at M lines, which are in the center of your sarcomere. So there's your muscle, there's your bundle of muscle fibers. You can see the individual muscle fiber cell, and then the myofibrils that are making that up. And you can see the overlap between the red and the blue. And then if we keep looking a little bit closer, you can see the M line is where your myosin, your thick filaments are lined up, while the actins are attached at the Z lines, and they are pointing towards that M line. So why muscles are able to contract, the model that we use to describe this is called the sliding filament molecule. The filaments, the actin and myosin, the thin filaments and the thick filaments are able to slide past each other longitudinally, creating an overlap. To be able to do this, the head of a myosin molecule will bind to an actin filament, forming a cross bridge and pulling that thin actin filament closer towards the center of your sarcomere. Muscle contraction will require multiple cycles of both binding and release. Glycolysis and aerobic respiration are able to provide the ATP energy that is needed to sustain this contraction. So there's your relaxed muscle, and then you have your contracting muscle. You can see the actin filaments are moving closer together. And then when you've reached a fully contracted muscle, your sarcomere is much, much smaller because it is has contracted as far as it can go. So in order for this all to happen, again, you've got your thin filaments and your thick filaments. There's your myosin head. You're going to need to use ATP to get that myosin head into its most energetic configuration. There are binding sites on the actin that will allow that high energy myosin head to be able to form a cross bridge with those actin filaments, and that will help to pull them closer together. Once they've made the cross bridge, the actin filament will then move towards the sarcomere center. Um, we don't want our muscles to contract all the time. So there are regulatory proteins, tropomyosin and troponin, which bind to actin strands on those thin filaments when a muscle fiber is at rest, so that the actin and myosin do not interact with one another. For them to be able to contract together, those binding sites for the myosin is able to bind to the actin have to be uncovered. This happens when calcium ions are able to bind to that troponin complex, and that will remove it from the myosin binding sites, making those exposed and allowing the myosin then to bind to it. So contraction will occur when you have high levels of calcium present. It will stop when your calcium concentrations are much lower. Okay, so there you can see the calcium binding sites and how the myosin binding sites are blocked. Adding all sorts of calcium is going to expose those binding sites by moving your troponin away from the troponin complex, away from the tropomyosin, or sorry, away from the actin filaments. And now you can have contraction take place. So, to have a contraction occur, a stimulus must happen that will result in an action potential that allows a motor neuron to synapse with a muscle fiber. When that takes place, the synaptic terminal of the motor neuron will release acetylcholine, that neurotransmitter we talked about previously, which will cause a depolarization in the muscle, leading it to produce an action potential. Those action potentials move to the muscle's interior fiber along transverse or T tubules. That action potential also will cause the sarcoplasmic reticulum to release calcium ions, which will expose the binding sites for myosin on the thin filaments on the actin. Okay, a lot of things have to take place to be able to get that contraction process to occur. We talked about how it's going to cause that cross bridge cycle to proceed. When the motor neuron input stops and you're not having those action potentials anymore, the muscle cell relaxes. Transport proteins will get calcium out of the cytoplasm and that are present in the sarcoplasmic reticulum. The proteins that were bound to the thin filament will shift back and block the myosin binding sites so that contraction no longer takes place. In amyotrophic lateral sclerosis, ALS, also known as Lou Gehrig's disease, there are issues with skeletal muscle fibers being excited properly. This typically is a fatal disorder. Myasthenia gravis is an autoimmune disease. It is going to attack your acetylcholine receptors on your muscle fibers. There are some treatments for this one. Contraction of a whole muscle is graded. So the extent of your contraction, the strength can be altered voluntarily by impacting the number of fibers that are contracted and the rate at which those fibers are stimulated. Vertebrates, each motor neuron is able to synapse with multiple muscle fibers, but each fiber is only controlled by one of those neurons. A motor unit will consist of that single motor neuron and all the fibers it's able to control. If multiple motor neurons are recruited together, then you will have a stronger contraction. A twitch will happen when you have a single action potential take place in a motor neuron. Again, we talked about summation previously. When you have many more action potentials delivered rapidly, they can produce a graded contraction via summation. Tetanus is, we're going to talk about the difference between this and the vaccine that you get. It's a state where you have smooth and sustained contractions because your motor neurons are delivering a volley of those action potentials to your skeletal muscles. So it's allowing things to continue to take place. The tetanus that you take a vaccine for is caused by bacterium Clostridium tetani, which makes toxins that interfere with those contractions. Okay, so there you have from your spinal cord, your nerves going out. You've got two motor units in this particular example. You will notice that there are different synaptic terminals connected to each of those nerves, but that there is only one synaptic terminal present on each muscle fiber. So there's a single twitch is the one action potential. If you have multiple twitches, you get them combined together, that summation process. And when things really get going and you've got lots of action potentials taking place, tetanus will result. There are different types of skeletal muscle fibers depending on their function. How we're able to distinguish between them depends on their source of ATP or by how quickly they contract. There are oxidative fibers that are going to rely on aerobic respiration to get you your ATP because they have lots of mitochondria, lots of blood, and lots of myoglobin present, which is able to help to bind the oxygen more tightly than hemoglobin. Then there are the glycolytic fibers, which are going to use glycolysis. Remember, that's not make as much ATP, not as many myoglobin are present, and they are going to tire more quickly. When we talk about light meat versus dark meat in both poultry and fish, the light meat is going to be primarily your glycolytic fibers, while dark meat is going to consist of your oxidative fibers. There are both slow twitch fibers and fast twitch fibers. Slow twitch fibers contract slower, but they are able to sustain longer contractions. They are always going to be oxidative. Fast twitch fibers will contract more quickly, but they are only able to sustain your shorter contractions. They can be either glycolytic or oxidative in terms of their source of ATP. Skeletal muscles will contain both of these types of fibers, but they'll have different ratios of them. Some vertebrates will have muscles that twitch rates a lot faster than humans. They can move faster than us. The male toadfish, we saw a picture of him on the previous slide, to produce his characteristic mating call, will contract and relax muscles more than 200 times per second. Cardiac muscles found only in the heart, striated cells that are connected by intercalated disc. We talked about and we discussed cardiac, that gap junctions are able to connect or to provide coupling between cells. Again, the left ventricle and the right ventricle, the left atrium and the right atrium. They're able to generate action potentials without neural input. That's why the heart can keep beating even if the brain has stopped. In smooth muscle, cells don't have striation. They contain less myosin. Myosin that is present is not attached to specific actin. We tend to find this in your hollow organs, like those that are found in your digestive tract. Their contractions are fairly slow. They're either initiated by the muscles themselves or they are able to be formed from stimulation of neurons that are present in the autonomic nervous system. Skeletal systems are able to use muscle contraction and turn it into locomotion. The muscles are attached in antagonistic pairs, so they work opposite one another. Their actions are coordinated by the nervous system. The skeleton provides a structure to which these muscles can attach, and it also helps provide support, protection, and movement. So there we see the biceps and triceps. Those would be antagonistic muscles. the extensor and the flexor muscle in the grasshopper tibia. And we'll talk about the different skeletons in a second. Three types of skeletons: hydrostatic, which lack hard parts; exoskeletons, which are found externally, they're hard portions; and endoskeletons are internal hard parts. Hydrostatic skeletons are fluid that is held under pressure in a closed body compartment. We find this in cnidarians, flatworms, nematodes, and annelids for the most part. Annelids are able to use this for peristalsis. It's a land movement that's produced by rhythmic waves of muscle contractions. Okay, so there you can see its particular movement. Exoskeletons, hard encasements that are on the surface of an animal. We find these in mollusks and arthropods. Arthropods have a jointed exoskeleton called a cuticle that can be both strong and flexible, and chitin, the polysaccharide, is often found in it. Endoskeletons are what humans have: hard internal skeleton that's buried in soft tissue. It's found not just in mammals, but in organisms ranging from sponges all the way up to mammals. The mammalian skeleton has over 200 bones. Some of these bones are fused, others are connected by ligaments, so that helps to improve motion. And so joints, you have your ball and socket joint, which is at your shoulder. We have our hinge joint, which would be at our elbow to connect our humerus and our ulna. And then we have our pivot joint, which is going to allow our hands to be able to move, our ulna and our radius. And there are some other examples of the specific bones making up the human skeleton and the joints. Size and scale of skeletons. You need to have a skeleton that will be able to support the size of that animal. The weight of the body will increase with the cube of its dimensions, but the strength increases with the square. So you can have a greater amount of weight. You will have a greater amount of strength, but not as proportional to the weight. Small and large animals will have different proportions for both mammals and birds. Your leg position will be critical to determining the weight that they can bear. Locomotion, we can talk about different types of ways that you can travel between places. You have to use up energy to overcome both friction and gravity. You can have locomotion on land, in water, or in air. On land, we have to be able to move against gravity. There are lots of adaptations that have taken place that have evolved over time in vertebrates. For water, we're dealing with swimming. Don't have gravity as much of an issue, but friction plays a role. So your faster swimmers are going to be more of a torpedo-like shape, sleek, so they can kind of minimize that friction. Ways animals can swim. They can paddle using their legs as oars, jet propulsion, or undulations, moving side to side and up and down. And then flying, locomotion in air. Wings have to have enough lift to overcome the downward force of gravity. Flying animals have adaptations that have helped to reduce their body mass so they can cut down on gravity. Examples include birds lacking teeth and a urinary bladder, and they have bones that have lots of air-filled regions. Locomotion and its energy costs. The amount of energy that you will need will depend on um, where you're going, how far you're moving, and what other factors have to play a role. We talked about gravity and friction in making that movement possible. We can estimate this using rates of oxygen consumption as well as carbon dioxide production. If you are specialized for swimming, you tend to spend less energy per meter traveled than animals of similar sizes for both flying and running. Larger animals are typically going to be more efficient than smaller animals for the same mode of transport.